Welcome to Unacademy. Welcome to Unacademy EAMCET live classes, students. So in today's class we are going to discuss about the cell cycle and cell division. That is the second part. So in last class we were discussing about the first part. In the second part we will also discuss about the cell cycle and cell division. So we'll be discussing the part 2. So in this So thank you, students. So let us start the topic. So in today's class we'll be discussing about the lesson that is cell cycle and cell division. So this is the part 2 of the cell cycle and cell division. So in this lesson, if you see this particular diagram, so this diagram is to explain that the cell cycle and cell division, this lesson. So in this diagram, if you clearly observe that what happens here, this is the testis. So this is the male reproductive part of the human being, that is called as a testis, and this is the male reproductive part of the woman, that is called as ovary. So from the testis, through a process that is inside the testis, so through a process that is called gametogenesis, so gametes are produced through, and the formation of gametes takes place through the process that is called as meiosis. So this clearly tells that. So today topic we are going to discuss about the meiosis. In last class we were discussing about the mitosis in the part 1. So in today's we'll be going to discuss about the meiosis. So due to gametogenesis, what happens is, during gametogenesis meiosis will take place, and formation of male gamete and formation of the female gametes will be taking place in the human beings. And during the process of fertilization, what happens, these two male gametes, so you can see this is the egg and this is the sperm, and see these two male gametes fuse, and that process is called as fertilization. And later after fertilization, what happens is the formation of the diploid zygote will be taking place, and the zygote develops into a new individual. So for all these to take place, the meiosis is the main step in the formation of the gamete. So that is the diagram is to show that meiosis is taking place, and it is the major important event in case of the human beings, especially in the males to produce the gametes, and as well as in females also to produce the gametes. So before starting the topic that is meiosis in detail, so let us see about our Unacademy Plus courses. So Unacademy, every day Unacademy is providing free live classes. At the same time, Unacademy is also producing the free live classes on YouTube, Unacademy EAMCET. So apart from that, Unacademy is also giving the plus courses. So that is called as for the paid subscribers. So in this, a designed course will be present. And there is a comprehensive batch which started recently in the September first week. So in that comprehensive batch, I will be taking the biology classes, and this sir will be taking the physics classes, and this sir will be taking the chemistry classes. So all are well experienced, having more than 20 years of experience, and this batch already started on the September 2nd, and this batch main purpose is for the students who are preparing for the next year medical entrance examination, that is EAMCET. So what is importance of this comprehensive batch? If you see, complete syllabus will be covered, and the tips and tricks also will be given by the top educators in case of the physics, chemistry and biology, and topic wise practice papers will be also made available by the respective educators, and full length mock tests will be conducted whenever and wherever required and wherever necessary for the students actually. So there is 1 month plan. So 1 month plan, if you are interested, you can take the 1 month plan, that is 2,250 rupees, and that is valid only up to 30th September. After that there is maybe an increase in the price also. So if you are interested, you can go to our website and open the link, and you can take the course. And for that, if you use the code, that is Rajendra Live, if you use my code Rajendra Live, you will get 10% discount on the particular courses. See, apart from the 1 month plan, so just now I am discussing about the 1 month plan. Apart from the 1 month plan, there is a long term plans are also there. So 6 months plan as well as the 12 months plan. So 6 months plan is of about, if you take the subscription for 6 months plan, it is costing about 10,125 rupees. And if you take 12 months plans, if you take 12 months plan, it is of 12,375 rupees. So 12 months plan is the best plan. If you see per 1 month, it is approximately, it is charging like 1,031 rupees. So for 1,031 rupees, biology, physics and chemistry all together will be coming in one plan. So it will certainly benefit for the students who are preparing for the next year medical entrance examination. So what is importance of, once again, so what is importance of this Unacademy Plus subscription is, so premium content. So no doubt the premium content, so best quality in the material will be given, and structural live course will be, means a design live course, always a design course will be there, so that the 2 years, both 2 years course will be like intermediate first year, intermediate second year, or 11th class or 12th class syllabus. Whole syllabus will be completed by the before the examinations. And after every three lectures, and fourth lecture will be having a doubt session, so in that student can interact with the lecturer and he can clear his own doubt session. So the doubt session will be there, and he can clear his own doubts. And quizzes also will be conducted at regular intervals. So like that you can also, so different batches will be there. So there are several educators on our platform, so you can also choose the batch of the educators which you are interested to study with them. So that is importance of Unacademy. So I suggest you to take the best plan, that is 12 months plan for 1 year. It is costing about 12,375 rupees. And if you are interested, you can use my code, that is Rajendra Live. So Rajendra Live is my code. If you use this code, you will get the discount of 10%. So you are going to get the discount of 10% on the above prescribed course. So that is about our Unacademy Plus subscription students. So let us start the topic, that is meiosis in detail. So let us start the meiosis, students. So let us start the meiosis in detail. So term meiosis, if you see the term meiosis, okay, term meiosis, if you see the term meiosis, so term meiosis was coined by the scientist, that is the name of the scientist is called as a Farmer and Moore. So Farmer and Moore are the two scientists who has coined the term meiosis. So meiosis term was coined by the name of the scientist, that is called as a Farmer and Moore. So meiosis is characterized. So what is importance of this meiosis? So if you see, so what is importance of this meiosis? So meiosis is characterized. So meiosis is characterized by two successive divisions. So what is happening in meiosis means two successive divisions will be taking place. That two successive divisions are nothing but they're called as meiosis 1 as well as the meiosis 2. So meiosis is characterized by two successive divisions. So in that, the division of cytoplasm is taking place, at the same time division of the nucleus is taking place. So two successive divisions, the name of the two successive divisions are called as meiosis 1 and as well as meiosis 2. During this successive division, the division of cytoplasm will be taking place, the division of the nucleus also will be taking place in case of the meiosis. So haploid cells, so these haploid cells, so whatever haploid cells either become or gives rise to gametes. So haploid cells which are produced through meiosis. So just now we have seen in diagram that male gametes, so male gametes from the male reproductive part of the animal, so they give rise to male gametes. Similarly, female gametes are also produced. So meiosis, what is the purpose of meiosis? So if you see the purpose of meiosis, so the meiosis ensures production of haploid phase in the life cycle of sexual reproducing organism. So generally sexual reproductive organisms, these are the higher organisms. In higher organisms, the cells are in diploid state. So in that they are always in diploid phase and the cells are of diploid. So haploid phase is also existing in the higher plants because of the meiosis, and that fertilization restores that particular diploid phase actually. So meiosis is required to run the reproductive cycles of the eukaryotes. So no doubt meiosis is essential at the same time to run the reproductive cycles of the eukaryotes. So that is importance of the meiosis. So what is the main purpose? If you see the main purpose of the meiosis. So what is the main purpose of meiosis here? To maintain, in case of humans, so main purpose of meiosis is to run the reproductive cycles of the eukaryotes. So welcome to students, welcome to Karishmika, welcome to Satvika, and welcome to few more students. So in this today's class we are discussing about the meiosis. Let us continue with meiosis, students. So meiosis involves two sequential stages. Okay, just now discussing, it is called meiosis 1 as well as meiosis 2. So in these two sequential stages, so the division of nucleus is taking place, at the same time division of the cytoplasm is also taking place. So in meiosis involve two sequential cycles of nuclear and cell division called as meiosis 1 and meiosis 2, but only one cycle of DNA replication. So what is the meaning of this particular sentence is to tell that. So this particular meaning of sentence is telling that the cytoplasm divides. So cytoplasm is dividing. So cytoplasm divides. Here cytoplasm divides twice. So what is the meaning of this sentence is twice. Cytoplasm divides twice, but the DNA divides only once. So that is important point you have to remember, students. In multiple choice question will ask you. So meiosis, what happens? In meiosis, what happens is during meiosis, cytoplasm will divide twice, whereas the DNA will divide only once. That is important point we have to remember here actually. So meiosis involves. So what happens in meiosis? Meiosis involves the pairing of homologous chromosomes. So in coming slides we will be seeing how the pairing of homologous chromosomes will be taking place and how the recombination will be taking place. So before that we are just seeing here actually. So in meiosis what happens? Pairing of homologous chromosomes and recombination. So recombination also will be taking place during the process of the meiosis. So no doubt at the end of meiosis what happens? No doubt haploid cells are formed at the end of the meiosis. So anyhow, so what happens in meiosis? In meiosis two cycles are there. It is meiosis 1 and meiosis 2. So during that process what happens is cytoplasm divides twice and DNA divides only once. This is the important point you have to remember, because this is generally routinely asked in the multiple choice point of view. Okay, now let us see about the diagram which represents the meiosis actually. So if you see, this is the meiosis. So it is having the meiosis always takes place. So meiosis always takes place in a diploid cell. That is another important point you have to remember. So meiosis always takes place in a diploid cell. So this diploid cell, so during interphase, so we already have discussed about the interphase. That interphase is having the three major stages like G1, S phase and G2. So that interphase is also present before meiosis 1. Interphase also will be taking place like G1 phase, S phase and G2 phase. These all phase will be taking place before the meiosis 1. So interphase is present in case of mitosis as well as in case of the meiosis also. So interphase will be present. So after interphase it is followed by the meiosis 1, and after meiosis 1 it is followed by the meiosis 2. So in meiosis 1, so at the end of meiosis 1 you will have the two dot cells, and two dot cells will be having the haploid nature. So haploid nature. So this is the cell which is diploid, and this is converted into haploid cell. So this piece is called as a, so at the end of meiosis what happened? Reductional division is taking place. So why it is called reductional division? Because here chromosome number is diploid. When it comes to the dot cells at the end of meiosis 1, the dot cells are haploid in nature. So reduction of the chromosome number is taking place. So that is the reason it is called as a reductional division. So meiosis 1 is called as a reductional division. So if you see in the again after meiosis 1, so meiosis 1 again undergoes division. Okay, so after meiosis 1 what happens? Cell will undergo cell division, that is called meiosis 2. So in meiosis 2 what happened? Again two dot cells will be produced at the end of meiosis 2, and these dot cells are haploid. So this is a equational division. So this is an equational division. So finally what we have to remember here is, so meiosis 1 is called as a reductional division, meiosis 2 is called as equational division. So why meiosis 1 is called as a reductional division? Because in meiosis 1 the chromosome number is initially diploid, and by end of the meiosis 1 chromosome number has been reduced. So reduced to haploid. So because of this reason it is called as a reductional division. So due to that reason it is called as a reductional division. So in meiosis 1, so we have different phases in meiosis 1 like prophase 1, metaphase 1, anaphase 1, telophase 1. So let us see about the prophase 1 of the meiosis. So let us see about the prophase 1 of the meiosis. And again in prophase 1 again meiosis, in prophase 1 of the meiosis, it is further classified into leptotin, zygotin, pachytin, diplotin and diacansis. So let us start with the leptotin. So in the prophase 1, let us start with the leptotin. So what I am telling here is, in meiosis, so meiosis, we already have seen the meiosis 1. So meiosis 1 having the prophase 1, it is having prophase 1, metaphase 1, anaphase 1 and telophase 1. So this prophase 1, so this prophase 1 is again classified into leptotin, zygotin, pachytin, leptotin, zygotin, pachytin, diplotin and diacansis. So prophase 1 is further classified into leptotin, zygotin, pachytin, diplotin and diacansis. Now let us see about the leptotin. So what is happening in the leptotin? So let us see what is happening in the leptotin of the prophase 1. Okay students, so let us continue with the leptotin 1. So let us see what is happening in the leptotin 1. Okay, so during this stage in leptotin, what happens? So chromosome gradually becomes visible. So chromosome gradually becomes visible under the light microscope. So actually what happens, if you see, so I will show you one diagram before going into the, so this is one of the diagram to explain you what is happening before starting of the meiosis 1. So this is the diagram, so before starting of the meiosis 1. So in interphase, if you see in interphase, what happens, you can see chromatin material. So chromatin material, so this chromatin material is disorganized, it is not organized, it is a disorganized. You are not able to see the chromatin material clearly, it is in a coiled manner, that's why it is called as a chromatin. It is a network like it is covering. So network like organized, so it is disorganized. So it is disorganized in case of the interphase 1. So when it comes to prophase, you can see in this diagram, when it comes to prophase, you are able to see that there is a chromosome. So condensation of the chromatin material takes place and it is organized now. The chromatin material is organized. You can see in prophase 1, chromatin material is organized in the form of a chromosome like structure. So like this one, chromosome is formed. So it is organized as a chromosome. So that's what we were discussing here actually. So what he was telling is, during this stage, in which stage? In the leptotin stage. In leptotin stage, what happens is, chromosomes gradually visible under the light microscope. So we cannot see the chromosomes with our naked eye. We have to use analytical technique. The analytical technique here we are using is a microscopy. So microscopy is an analytical technique which we are using to identify. We can see the chromosomes. So during this stage, what happens? Chromosomes gradually visible under the microscope. So in interphase, what happens? We are not able to see the chromosomes, but when it entered into leptotin, we can see some chromosomes under the microscope. So that's what he meant to say. So the compaction. So compaction of chromosome continues throughout the leptotin. So what is this? Compaction. So compaction means nothing but it is the condensation of the chromatin material. So that is called as a compaction, condensation of the chromatin material. So condensation of the chromatin material. So that is called as a compaction. So compaction of the chromosome continues throughout the leptotin. So condensation of chromatin material gives rise to one type of the chromosome. Okay. So the chromosomes at this stage take up specific covering towards one end of the nucleus where the centromere lies, and this is called as a bouquet stage. So he is telling that so chromosomes at this stage, okay, chromosomes at this stage take a specific converge towards one side of the nucleus. So what is happening here is, so I will show you one diagram. So in leptotin what happens is the nuclear membrane will be present. So nuclear membrane will not disappear. So like this. So at one end of the nuclear membrane, so chromosomes will be attached. The chromosomes will be attached to the one end of the chromosome. So chromosomes, okay. So chromosomes will be attached to the one end of the nuclear membrane. So like this. This is the centromere. Okay. These are this. So now this appearance is called as this appearance. So all chromosomes are attached to the one end. Here at this end the chromosomes are attached. So that is called as a bouquet stage. So it is also called as a Darlington. It is also called as a Darlington stage. So bouquet stage has another name. It is also called as a Darlington stage. So what is happening here? Chromosomes at this stage take up specific converge towards one side of the nucleus. So it is moving towards one side of the nucleus or not? It is coming and attaching to the one side of the nuclear membrane. So because of that, what happens? It appears like a flower bouquet. So it appears like a flower bouquet. So that is the reason it is called as a bouquet stage. And bouquet stage was first identified by a scientist. Name of the scientist is called as a Darlington. So that is the reason this particular stage is called as a Darlington stage. So bouquet stage is called as a Darlington stage. So in leptotin what happens? So you can see here this particular diagram. If you see this diagram you can clearly understand. So you can see the chromosome here. So you can see how the chromosomes are dispersed. Okay, here also you can see chromosome is clearly seen the centromere at the center and chromatids. So you can see the chromatids with centromere at the center and chromatids. So like this in the leptotin stage. So what happens is chromosome appears, okay, with centromere at the center and chromatids are clearly observed. Okay, see the centrioles duplicate. So during leptotin stage what happens? Centrioles are also will be duplicating. So what is the main purpose of centrioles? The main purpose of centrioles is for the formation of spindle fibers. Centrioles main purpose is for the formation of spindle fibers. So spindle fibers. So for the formation of spindle fibers, centrioles also will be duplicating, and each daughter centriole migrates towards the opposite poles. So daughter centrioles also will be migrating towards the opposite poles. So what I was telling here is centrioles also will be duplicating. So I will be showing in diagram how centrioles is duplicating. So let us see this diagram. So once again, so you can see here these are the centrioles. This is actually called as mitotic spindle. So you can see here centrioles are also dividing in the prophase 1. Okay, in the leptotin stage what happens? These are the centrioles. So centrioles are also undergoing the division. So what is the main purpose of centrioles in the metaphase? When it comes to metaphase 1, what happens? Centrioles develop into spindle fiber like structure. So that is the main purpose of this particular spindle fibers. Means the main purpose of the centrioles is to produce the spindle fibers. Okay. So on reaching the poles, each centriole duplicate again, and at each pole possesses 2 centrioles. So each pole is possessing the 2 centrioles, or it is called as a diplosome. Next, that is about the leptotin stage, students. So finally, what is happening in the leptotin stage? So in leptotin stage, what happens is the condensation of the chromosome is taking place. Then what is happening? Then the formation of the Darlington stage or bouquet stage is observed in the leptotin stage. So, and centrioles are migrating towards the opposite poles, and they are ready to form the spindle fibers, and they are forming the mitotic spindle actually. So next one is zygote stage. So next stage is called as a zygote stage. So leptotin stage is followed by the zygote stage. In this stage, so what is happening in zygote stage? Pairing of the homologous chromosomes is taking place. So this is the important point you have to remember. So pairing of the homologous chromosomes is taking place in case of the zygote in stage. So what is that pairing of homologous chromosomes means? For example, this is one chromosome. So this is one chromosome and this is one chromosome. This is from the, think like this is from the mother, and this is another chromosome. This is from the father. So these two are called as a homologous chromosomes. So these two are the homologous chromosomes. Now these two homologous chromosomes, what they are doing? They are undergoing the pairing. So what is happening? Pairing. So what is the pairing? So like this. So pairing is taking place. So now this is called as a pairing, and in between this there is some proteins will be present, and this all together is called as synaptonemal complex. This all together is called as a synaptonemal complex. Now this is called as synapses. So at this stage, what is happening at this stage means the pairing of the homologous chromosome is taking place. At this stage, what is happening is pairing of the homologous chromosome is taking place at this stage actually. So pairing of the homologous chromosomes is taking place. So in homologous chromosomes, so what is happening in this homologous chromosomes? So one chromosome is coming from the mother. So that's what I have written here. One chromosome is coming from the mother, and the one from the father. So one chromosome is from the father. So they are attracted to each other and pairing will be taking place. So they are attracted to each other and the pairing is taking place. So that pairing is called as a synapsis. And these two, so now these two homologous chromosomes which undergo the synapsis, and these are called as bivalents. So in multiple choice question they will ask you the formation of bivalents. The formation of bivalents takes place in which stage? Like that they will ask in multiple choice question. The formation of bivalents takes place in case of the zygote. So anyhow, so what is the important event that is taking place in the zygote means during the zygote stage, what happens is pairing of the homologous chromosome is taking place. So what is this homologous chromosomes? These are the chromosomes. In that homologous chromosome, one chromosome will be coming from the father and one chromosome will be coming from the mother, and these two chromosomes are involved in the process of the pairing. So what is the purpose of this pairing? So they will undergo the exchange of genetic material during the process of the meiosis. For that reason they start undergoing the pairing. So one pair of homologous chromosomes is called as a bivalent. So you have to remember this. One pair of homologous chromosomes is called as a bivalent. So just now I am telling that there is a formation of the synaptonemal complex. Now this is called as a synaptonemal complex. So let us see about the synaptonemal complex. So you can see in this particular diagram also. So this particular diagram also is showing how the synaptonemal complex is taking place. So these are the homologous chromosome. This is one homologous chromosome from one parent, and this is from the another parent. So one can from the mother and another can from the father. So what is the synaptonemal complex? So the pairing of homologous chromosomes. So pairing of homologous chromosomes is very specific. Okay, and it brings the alleles. So it brings the alleles of the homologous chromosomes exactly to the opposite to each other. So it brings the alleles. So means for example this homologous chromosome, if it is the, in humans we have 23 pairs. We have 23 pairs of the chromosomes in humans. If this homologous chromosome is of 5th chromosome, if this is the 5th chromosome of the father, then another one, this also should be the 5th chromosome of the father only. This also should be the 5th chromosome of the father only. For example, if this chromosome, this homologous chromosome is of the 5th chromosome of the mother, then this homologous chromosome should be 5th chromosome of the father. So like that. So that is the reason it is clearly mentioned that it is specific. It is specific. What is this meaning of the specific means? That homologous chromosome should be of the having the specific, like if the one of the homologous chromosome is from the 5th chromosome, the other homologous chromosome is also of the 5th chromosome of the different parent. So like that. So Moses is the name of the person who has discovered the synaptonemal complex. Okay, so which is paired homologous chromosome held by a nuclear protein. So synaptonemal complex. So this synaptonemal complex, so just now I have shown you. So this synaptonemal complex is having this protein at the center. So this center, this protein, so this is nothing but it is one protein. So what type of protein here? It is a nucleoprotein. So nucleoprotein. So what is synaptonemal complex? In multiple choice question they'll ask you. So what is synaptonemal complex? What is synaptonemal complex means? You have to clearly remember that two homologous chromosomes plus nucleoprotein. So two homologous chromosomes plus nucleoprotein together is called as a synaptonemal complex. So that is how we have to remember. So what is synaptonemal complex means? Synaptonemal complex is composed of two homologous chromosomes. It is made up of two homologous chromosomes and nucleoprotein. So two homologous chromosomes along with the nucleoprotein is called as a synaptonemal complex. So that is about the synaptonemal complex. So how the synaptonemal complex is formed? Let us see how the synaptonemal complex is formed. So complex extends along the whole length of the paired chromosome and is usually anchored at either end of the nuclear envelope. Okay, so it is extending whole the length of the chromosomes. Whole length of the chromosome is involved actually. If you see this diagram, you can see. So this one here, it is shown here only this part. So what he was telling is whole the length of the chromosome up to here. So up to here the pairing will be taking place actually. He used to say this part and this part will be attached like this. Whole the length of the chromosome will be associated. Like for example, I will draw one diagram. So this is one chromosome, this is another chromosome, like this. Whole length of pairing, yes, like this, except at the centromere. See, so whole length is pairing or not, like this. So that is what the particular meaning is, whole length of pairing. Okay, so whole length of the paired chromosomes and is usually anchored on either side of the nuclear envelope. So it is located on either side, means it can be towards one side of the nuclear envelope or other side of the nuclear envelope. Next, synaptonemal pairing produces an opportunity for crossing over. So what is the purpose of the synaptonemal complexes? In the next stage, after completion of the packaging stage, after completion of zygote stage, so cell will enter into packaging stage. In the packaging stage, what happens? Crossing over will be taking place. So it will be assisting. So synaptonemal complex will assist the homologous chromosomes for the crossing over. So during that is occurring in the packaging stage. So the pair of homologous chromosomes is called as a bivalent. So just now we have seen the pair of homologous chromosomes. So just now we have seen the pair of homologous chromosomes. So this pair of homologous chromosomes is called as a bivalent. We have seen this pair of homologous chromosomes. So this one. So this pair of homologous chromosomes is called as a bivalent. So that is about the zygote stage. So finally, what is happening in zygote? So what is happening in zygote means it is nothing but pairing of the homologous chromosome is taking place in case of the zygote, and formation of synaptonemal complex is taking place. So what is synaptonemal complex is composed of? Synaptonemal complex is composed of homologous chromosomes as well as nuclear protein. So that is what happening in the zygote stage. So let us see about the next stage, that is called as a packaging stage. So what is happening in packaging stage? Let us see what is happening in the case of the packaging stage. So in packaging stage, what happens? This stage is characterized by the appearance of the recombinant nodules. So this stage is characterized by the appearance of the recombinant nodules. So what is happening here is this stage is characterized by appearance of the recombinant nodule. So there is a formation of recombinant nodule is taking place. So the sites where crossing over occur between the non-sister chromatids, the sites at which crossing over occurs between the non-sister chromatids. So non-sister chromatids. So crossing over is the exchange of the. So what is the crossing over? It is the exchange of the genetic material between the two homologous chromosomes. So see what is happening in the packaging. That's what he was telling here is. So when cell enter into packaging stage, what happens here is, when cell enter into packaging stage, the chromosome appears in this manner, in this manner. So previously we have seen chromosome of this type. Now chromosome with the two chromatids can be clearly seen in the case of the packaging stage. Now like this chromosome is appear. Now like this it will be appearing. Okay. Now this is one chromatid, this is one chromatid, it is having this DNA, this is one chromatid. So two chromatids. This chromosome is having how many chromatids? So it is having the two chromatids. So this is one chromatid and this is the another chromatid. So two chromatids it is having. So now what happens is crossing over, exchange of genetic material taking place. So exchange of the genetic material is taking place between the two homologous chromosomes. This chromosome is from the mother and this chromosome is from the father. So what I am telling is, previously in case this is in the packaging stage. In zygoting stage we have seen chromosome appear like this, but when it comes to the packaging stage, chromosome appears in this way. Okay, so like this. So exchange of the genetic material. So in crossing over, what happens here is exchange of the genetic material. So exchange of genetic material will be taking place. So this the exchange of genetic material taking place in the crossing over. So at the same time, what he was telling that crossing over occurs between the non-sister chromatids of the homologous chromosome. Now for example, this is one chromatid and this is another chromatid. On the same chromosome, two chromatids are present. On the same chromosome, two chromatids are present. These are called as sister chromatids. These are called as sister chromatids. So on the same chromosome, if two chromatids are present, this is one chromatid and this is another chromatid. So two chromatids are present. Those are called as a sister chromatids. So here, this is different chromosome from the father. This is the different chromosome from the mother. So the exchange of genetic material is taking place between the one chromatid of the mother and other chromatid of the father. So those two chromatids are called as non-sister chromatids. Those two chromatids are called as a non-sister chromatids. So you have to remember this. In multiple choice questions they'll ask you, what is happening in the process of the crossing over? In the process of crossing over, what happens is exchange of genetic material. So exchange of the genetic material is taking place between the homologous chromosomes. So exchange of genetic material is taking place between the homologous chromosomes. So exchange is taking place, no doubt. Exchange is taking place between what type of chromatids? Exchange is taking place between the non-sister chromatids. Exchange is taking place between the non-sister chromatids actually. So in a chromosome, if you see this chromosome, every chromosome has two chromatids. So this is called as one chromatid. So like this, all together, this is one chromatid and this is another chromatid. So every chromosome has two chromatids, and these two chromatids are called as sister chromatids. So this chromosome is from the mother and this chromosome is from the father. When pairing is taking place between the chromatids of mother and with the chromatids of the father, that type of exchange is taking place between non-sister chromatids. That is called as a non-sister chromatids. So you can see in this particular diagram like how the crossing over is taking place, how the crossing over is taking place between the bivalents. You can see in this particular diagram. So during this crossing over, so just now we were telling that there is a crossing over is taking place. So this crossing over, so what is the crossing over? Exchange of the genetic material is taking place. That is called as a crossing over. So during the exchange of the genetic material, there is one enzyme is used. The name of the enzyme is called as a recombinase. So enzyme that is used in the recombination. So that exchange of genetic material is called as recombination. So exchange of genetic material is called as a recombination. So recombination takes place with help of enzyme. The name of the enzyme is called as a recombinase. So for example, let us see once again about the crossing over diagram only. So this is the diagram. So like this, crossing over is taking place. So this is the site where the crossing over is taking place. So now this is called as recombination nodule. So this process, this is called as recombination nodule. So Here the recombination is taking place. So that recombination is assisted. So recombination is assisted by enzyme that is called as recombinase. So due to recombinase activity, what happens? This side fragment. So this side fragment will be moving towards this side, and this side fragment will be moving towards this side. Because of that, what happens is genetic material exchange will be taking place. Because of that, what happens is genetic material exchange. So genetic material exchange will be taking place. So because of that, genetic material exchange will be taking place. So because of that, what happens is genetic material exchange will be taking place. So that is supported by enzyme. The name of the enzyme is called as a recombinase. Next, so crossing over leads to recombination between the homologous chromosome. So crossing over is nothing but leads to recombination between the homologous chromosomes, and it is completed at the end of the packaging. So finally what we have to remember, students, what is happening in packaging stage. So what is happening in packaging stage? The main events in the packaging stage is nothing but crossing over is taking place. Okay, so crossing over is occurring between the non-sister chromatids. So crossing over is occurring between the non-sister chromatids of homologous chromosomes. So non-sister chromatids are homologous chromosomes. Then what is the enzyme involved in the packaging? Okay, the enzyme involved is called as a recombinase. The enzyme involved in the packaging stage, the enzyme involved in the recombination is recombinase. So the exchange of genetic material, is also called as recombination. So recombinase is the enzyme. Then what is formed during the recombination? The formation of the recombinant nodule. So they'll ask in multiple choice questions. Recombinant nodule. So recombinant nodule is formed in which stage? Recombinant nodule is formed in the packaging stage. In the packaging stage, recombinant nodule is formed. So just now we are telling that the enzyme involved in recombination is no doubt the enzyme involved in genetic material exchange, or the enzyme involved in the recombination is recombinase. Apart from recombinase, there is other enzyme is also involved. That enzyme is called as a ligase. The enzyme is also involved in ligase. So what are the enzymes involved in the crossing over means we have to tell that recombinase as well as ligase. And formation of recombinant nodule is taking place during which stage, they'll ask you. Then we have to remember it is in the packaging stage. So like that we have to remember about the packaging stage. So what is the major event is crossing over is the major event that is taking place in case of the packaging stage. So let us go to the next stage, that is called as the diploting stage. So at diploting stage, so in diploting stage what happens? So further thickening and shortening of the chromosomes takes place. So what happens is further thickening and shortening of the chromosomes is taking place. So you can see that this is the synaptonemal complex. You can see here still synaptonemal complex is existing in the diploting stage also. So the synaptic forces between the homologous chromosomes come to end. So in the diploting stage what happens is synaptonemal complex slowly starts disappearing. So the synaptonemal complex appears to be dissolved. So in multiple choice question they will ask you synaptonemal complex dissociates. Synaptonemal complex dissociates in which stage? They will ask in multiple choice questions. You have to remember that it is in the diploting stage what happens is synaptonemal complex gets dissociates. So in diploting stage what happens? So after crossing over the chromosomes, due to crossing over, genetic material exchange will be taking place. Because of that, the chromosomes appear X-shaped. So that is important point you have to remember. That X-shaped chromosomes is called as a chiasmata. So that X-shaped chromosomes is called as a chiasmata. It is called as a chiasmata. So this diploting stage, so this prolonged diploting stage, what happens is the prolonged diploting stage means the diploting stage keep on continuing for months and years. If the diploting stage keep on continuing for months and years, what happens is there is a formation of the giant chromosomes will be taking place. So there is a formation of the giant chromosomes. will be taking place. So So there is a formation of joint chromosomes will be taking place, and it is called as a lamp brush chromosomes. And these lamp brush chromosomes are observed in the oocytes of the some vertebrates. So in multiple choice question they'll ask you. So the prolonged diplotin stage leads to formation of the lamp brush chromosomes. That is important point which you have to remember. The prolonged diplotin stage leads to formation of the lamp brush chromosomes. So after that what happens? So nucleolus is diminished. So nucleus also will be diminished, but still it is persist. So no nuclear membrane. So nuclear membrane also disappears by the time diplotin stage occurs. So that is about the diplotin stage. Finally, what you have to remember in diplotin stage is chromosomes appear X-shaped, and that X-shaped chromosomes are called as a chiasmata. That X-shaped chromosomes is called as a chiasmata. And prolonged diplotin stage leads to formation of the lamp brush chromosomes. That is important point which we have to remember. So that is in the diplotin stage. Next one is diakinesis. So what happens in diakinesis? So at this stage, so bivalent chromosomes become more condensed. So the bivalent chromosomes, what happens? It becomes more condensed. You can see the chromosomes have become more condensed. So nucleolus detaches from the nuclear organism and ultimately disappears. So finally what happens is, by end of the prophase, nucleolus and nuclear membrane both will be disappear. So already nuclear envelope will be breaking down and nucleus also will be disappears. So what is the major event in diakinesis if you remember? So terminalization. So the terminalization will be taking place. If you see what is this terminalization means, so if you see here in this particular diagram, so this is the synapse. During synapse what happens? See, the chromosomes are attached in a full length like this during the synaptonemal complex. Now what happens is, the chromosome will be attached only at the ends. Chromosome will be attached only at the ends. Such process is called as a terminalization. So terminalization takes place. So terminalization takes place in case of the diakinesis. So that's what you have to remember in multiple choice question they ask students. What is the important event? So what is the important event in case of the diakinesis means you have to remember the terminalization. So terminalization will be taking place. Terminalization. So what is this terminalization means? So just now I am showing you in the diagram. Again I am drawing this. So like this chromosome will be there. Only chromosome after crossing over is over, synaptonemal complex breaks, but still the ends of the chromosomes are attached. So ends of the chromosomes are attached. Such process is called as a terminalization. Such process is called as a terminalization actually. Now let us see this diagram in detail actually. So that will conclude this session. So let us see this diagram clearly. So in the meiosis 1, so in meiosis 1 we have interphase, no doubt about it, we have interphase. So like mitosis, here also interphase will be present. So in interphase what happens, you can see chromatin material, and chromatin material is not organized. So it is not organized, it is a disorganized. Chromatin material is disorganized, and chromatin material appears as a thin filament like structure and it is disorganized. In the cytoplasm you can see the centrosome is present. Inside centrosome you can see centrioles. In each centrosome, we have one pair of centrioles. So you can see there is one pair of centrioles are present. One pair of centrioles are present inside the centrosome. So now this interphase, this is the interphase cell in the interphase. Now the cell will enter into the meiosis 1, that is in the prophase 1. So in the prophase 1, just now we have seen in the prophase 1, in the very first stage there is a formation of bouquet stage. After that bouquet stage what happens, in zygote synapses will be taking place. So you can see here also after synapses, in packeting stage crossing over is taking place. You can see crossing over is taking place in this particular diagram. The crossing over is taking place. So blue color one chromosome is present. No, this is one homologous chromosome, and another, the red color another one. This is another homologous chromosome. So both are undergoing. Both are seen. That all together, those two homologous chromosomes are called as a tetrad. So that is called as a tetrad. So what is tetrad means? So like this, if chromosome appears, so this is called as a tetrad. Now this is called as a tetrad. Then what is bivalent? So when chromosome appears like this, now this is called as a bivalent. So now this is called as a tetrad. Tetrad is observed in which stage? Tetrad is seen in the packeting stage. So in multiple choice question they'll ask you, tetrad is seen in which stage? That is seen in the packaging stage. So bivalents are seen in which stage? Means bivalents are seen in the zygote stage. So in this diagram also you can see, now this is called as a tetrad. So here what happens is crossing over is taking place in these two homologous chromosomes. Crossing over is taking place. Similarly, you can see in this pair of chromosomes also. Here, especially here you can see. So here also there is a crossing over is taking place. So in this cytoplasm, if you see in the cytoplasm, the centrioles. So centrioles are producing the spindle fibers, and that the division of dividing the centrioles dividing, and they form a mitotic spindle-like structures. So after that, what happens? After that, they gives rise to the cell will enter into metaphase 1. In metaphase 1, what happens here is, so spindle fibers are well developed. So you can see in the cytoplasm. So spindle fibers are well developed. Centrioles move towards the opposite poles, and they produce the well developed spindle fibers. So these spindle fibers are attaching to the chromosomes. So spindle fibers are attached to this particular chromosome. So how they attach means, so like this. We have seen just now terminalization. The chromosomes are attached at the ends, at the end of the diplotin. So like this. The spindle fibers are attached to the chromosomes in this way. Spindle fibers are attached to the chromosomes in this way towards the opposite poles. So like this. So they are attached to the ends actually. So this, like this chromosomes. So chromosomes are located at the center, at the equator, or at the center in case of the, in the metaphase. So the spindle fibers are attached to the chromosome at a specific location. Spindle fibers are attached to the chromosome at a specific location. That location is called as a kinector core. So spindle fibers are attached to the chromosomes at a specific location. That specific location is called as a kinector core. So what happens in metaphase? In metaphase, what happens is the homologous chromosomes. So one from the mother and another from the father. One from the mother and one from the father, which are attached to the ends. And that process called terminalization. That chromosomes are, that homologous chromosomes. These two are the homologous chromosomes. These homologous chromosomes are on the equator. In case of the metaphase, what happens? They are at the center or at the equator, and they are attached by the spindle fibers from the opposite poles. And these spindle fibers are produced from the centrioles, and spindle fibers attached to the chromosome at specific location. The spindle fibers attached to chromosome at specific location, that is called as a kinector core. So after that metaphase 1, cell will enter into the anaphase 1. So cell will enter into the anaphase 1. After the metaphase 1, cell will enter into the anaphase 1. So in anaphase 1, what happens in anaphase 1? So we clearly see that in anaphase 1, so disjunction is taking place. What is the major event in case of the anaphase 1 means? So the major event, that is called as disjunction. So what is the disjunction if you see? See, just now we have seen, so this is the chromosome, like this homologous chromosomes are attached by thread-like structure called as the spindle fibers. Now the chromosomes get separated. Now see, chromosomes get separated. So now they are attached here, they are attached in the metaphase. Now when it comes to the anaphase, what happens? They get separated. Now this is separate and now this is separate. Now this separation is called as disjunction. Now this process is called as a disjunction. Now this process is called as a disjunction. So disjunction. So disjunction of the disjunction will be taking place in the anaphase 1. Okay, disjunction will be taking place in case of the anaphase 1. So disjunction of the homologous chromosomes takes place in case of the anaphase 1. So that is the major thing about the anaphase 1. So that is about the anaphase 1. So that is the end of this session, students.